Today I will be talking about Erwin Chargaff and the double helix. Chargaff was born in August of 1905 in what is now Ukraine. By the time he was 9, his family had relocated to Vienna and World War I had started. His family was on vacation and they decided to not go back to embrace the war, and they moved with their belongings to Vienna. He was very smart and he graduated the top of his class, which his parents worried about because he was a late person to speak. He eventually overcame that, learning and mastering 15 different languages. But he graduated, and at 18, he graduated a prestigious uh, high school, and he was unsure what he wanted to be. He ultimately chose to study chemistry because it was best chance at a stable job since his uncle owned a alcohol shop and he knew nothing about it, so he figured it would be a good test. He spent 5 years studying at the University of Vienna, and in the summer of 1928, he graduated with a PhD in chemistry. The next step in his life sent him to Yale in 1928. He was held up and arrested until Treat Johnson, one of the two scientists he worked with at Yale, caught him out because he was a student. Uh, Ralph Anderson and Treat Johnson worked with him, and over the 2 years that he was at Yale, he published 7 papers and discovered and studied many different things. Those are just two of the main things. In 1930, he and his wife became homesick, and they returned to Europe. After a while, he wanted change, and he eventually ended up in Berlin, which he left due to Adolf Hitler's uprising, Paris, which he left due to his family being homesick once again for America, and then he returned to New York. This is when he became aware of Oswald Avery from the New York Rockefeller Institute and his studies. Avery proposed that genes were made of DNA. While criticized by most scientists at the time, Chargaff decided that his studies had some basis. Working with several colleagues, he got interesting results based on DNA in 1947. He had went ahead and realized that different species handed down different DNA. His colleagues and himself had created a Mobius strip, which he believed could split apart at the middle and give two halves to the offspring to inherit. A Mobius strip, to him, was like a disc with one side raised, in which it would cut the disc in half and give the inner portion to one child and the outer portion to another. His first key discovery of his two that eventually become rules was in 1949. He found the proportions of bases in DNA depended on the species the DNA comes from. He also said that the number of possible arrangements of bases was truly enormous, and therefore DNA could very well be the agent of inheritance. His second major discovery, and his most vital one, was that DNA of any source for the amount of T was equal to A and the amount of C was equal to G, and so that they have a 1-to-1 ratio for each T to A and for each C to G. Other ratios, such as A to G or C to T, vary based on what species and what other DNA markers the species have. Cambridge University is where Chargaff, Watson, and Crick met. Watson and Crick had worked together beforehand, and Chargaff introduced himself to them informally and told him much about his rules and what he had discovered, along with a couple other theories he had at the time. It is said that Chargaff told them about his Mobius design, and that Crick deduced that the bases were pairs, using his second rule and his original design. This led to further research and the discovery of a lock and key replication system. So Crick decided that each individual pair, or ratio of 1-to-1, held on to each other until they broke apart, giving way to new formations in DNA. On February 28, 1953, Crick and Watson determined that the structure of DNA was a double helix polymer, or a spiral of two DNA strands, each containing long chains of nucleotides wound around each other. That meant, within their model of DNA, they replicated themselves by not splitting away from each other directly and never coming back, but they unwound, and the A to T's or the C to G's created a new double helix on each side, and they would connect with their new replicated DNA strands. Watson and Crick's solution was formally announced on April 25th, 1953, following its publication in that month's issue of Nature magazine. Nature magazine was a popular scientific magazine around this time, and it revolutionized the study of biology and medicine. Some of the developments that followed directly from it, some of the developments that followed directly from it were prenatal screening for disease genes, genetic engineered foods, the ability to identify human remains, the rational design of treatments for disease such as AIDS, and the accurate testing of physical evidence in order to convict or exonerate criminals. The double helix discovery was so big that in most aspects it changed the face of genetics and the way the scientific community approached things of genetic quality. Because before this, genetics was looked at as this is what we have, this is what we know, we can't move forward from here because we don't know the even more basic point past genes. Once they realized that DNA and the double helix were the most basic point of genetics, they were able to find out so much more. And these are my citations.